Hello, everyone. It's my pleasure today to present for the Bridge.ai CM4AI internships the image data or IF module of the Bridge.ai CM4AI project. And this is done in the Lundberg lab. My name is Jan. I'm a postdoc in the Lundberg lab, and I'll guide you through what our lab does and how we contribute to the CM4AI project. So the Lundberg lab is interested in measuring and modeling the spatiotemporal subcellular organization of the human proteome, because we believe that this will allow us to understand how proteins and their variations contribute to cellular functions and disease. So the Lundberg lab is part of the Human Protein Atlas, and this is the technique that we mostly apply, spatial proteomics with antibodies. And the Human Protein Atlas project has run now over almost 20 years, and in this project we've generated over 40,000 polyclonal antibodies that allow us to map almost any human protein in images. And over time, the Protein Atlas has generated over 20 million images of human cells and tissues, and these allow us to see which protein is where in the cell under which condition. So in the subcellular section of the Human Protein Atlas, which we are part of, we have always mapped proteins of interest in cells by using markers for different cellular structures, like the nucleus that we see here, the microtubules, and the ER, but also using one of these over 40,000 antibodies to map a protein of interest. So we have a lot of data with a protein of interest stained and these three reference markers. And an interesting phenomenon that we see here is that, for example, proteins are found in multiple cellular compartments. So here you see a staining of the protein in the nucleus, but also in the plasma membrane. On the right, we see variation of expression, so heterogeneity of the protein in the nucleus with weak staining and strong staining. There's a lot of more phenomena that we've described, and one of the key publications was in 2017, where we published the subcellular map of the human proteome, where we showed over 12,000 proteins and where they localize in different cells. And here you can just get a feeling for all these different subcellular structures that make up the cell and where proteins localize, like the nucleus that we've just seen, but also nucleoli or cell junctions, the plasma membrane, but we also see here the centrosome and many more structures. So the interesting part is that the phenomenon that I just described, like multilocalization and pleiotropy, is quite common. So over 50% of all proteins localize to more than one subcellular compartment. And they have been described in the context of moonlighting. So what does moonlighting mean? Here we have a very good example for moonlighting. This is enolase 1, and it has different functions in different subcellular compartments. It's a glycolytic enzyme in the cytosol, plasminogen receptor on the plasma membrane, and the tumor suppressor in the nucleus. So this multilocalization that we've seen may likely relate to a lot of heterogeneity in the functions that the proteins conduct in the different cell types. And just recently, we've also looked into another factor driving this pleiotropy, and that is metabolic state variations. So here we see protein that localizes to vesicles or the nucleoplasm. And we see that for metabolic state variations, the pleiotropy and heterogeneity is particularly high. So 40% of all the enzymes show single-cell heterogeneity. This can even be related to a certain metabolic function. For example, here we see two enzymes from a TCA cycle, and they are equally varied across cells, which shows that basically whole pathways are heterogeneous among cell populations. So we can also express this in the classical map of all biochemical enzyme reactions. What you see here is stable enzyme and variable enzymes, or enzymes with variable expression. You can see that there's a really substantial portion of reactions in the cell that are driven by variable expression or that would underlie these variations. And we conclude that metabolic enzymes spatiotemporally partitioning establishes non-genetic dynamic metabolic states, so that basically in this information on the protein level about varying protein levels, there's even much more determined information for the cellular state than on the genetic expression level. And if you are more interested in this data, you can check out the new section in the Protein Atlas version 24 and our preprint on this part. But what we actually want to talk about is proteome heterogeneity. And we've just been talking about the cell metabolism part, but there's also much more relationships that we investigate, which can be the cell cycle. The cell cycle has a significant effect on many proteins. Cell shapes may influence them. And we also try to study cell migration and signaling in relation to cellular protein heterogeneity. And here the CM4AI project comes into with important other functions that we try to study in light of heterogeneity, because in the CM4AI project, we will study perturbations and differentiation processes. So our contribution to the CM4AI project is to generate data and tools that allow us to visualize with subcellular spatial proteomics where proteins localize in cells, for example, in these untreated or anti-cancer treated breast cancer cell line, or how they basically relocalize and change differentiation processes. So a key data set or first data set that we've acquired here is based on this MDA triple negative breast cancer cell line. And this is the data set that you can work with today. And here we have characterized untreated cells, paclitaxel treated cells, and vorinostat treated cells. And you can, in the first glimpse, just seeing the cell markers, nuclei, microtubuli in the air, already see that this coincides with definitely big changes in the cell. For example, in paclitaxel treated cells, we see much more multinucleation, which relates to that this is a microtubule stabilizer. Whereas in vorinostat cells, the cells seem to get a little bit bigger, and you can see a little bit more membrane ruffling here at the edges. Now, when we look into the protein localization of the MAP protein, I can show you a few examples. For example, here we see TRIM24, which localizes the nucleoplasm in all three conditions. But we also see not enzymes, but this is an enzyme and other proteins that significantly change their location. For example, HDAC8, it's mostly in the cytoplasm in untreated cells, and then we find it only in the nucleoplasm of paclitaxel treated cells. And we also see it more like in untreated conditions in vorinostat treated cells. Oh, here we have a protein DNMT1, which just shows large spatial-temporal heterogeneity, or we assume spatial-temporal heterogeneity based on the intensity variation that we see in the three conditions. And there might be changes here as well. So how do we analyze such images and how do we integrate them into the CM4AI project? So there's a couple of challenges that when we analyze images appear. And first of all, there's a class imbalance. So there are very common patterns like the cytosol, and there are very rare patterns like the centrosome. At the same time, we see large morphological differences in this cancer data set that might be not such to large extent as we see here, but also as I have just described before, we see how the nucleus structure and the cellular structures change. Then we have single cell variation that we've just seen also in the cancer cells, and we have this multi-localizing aspect that make it really challenging to analyze these images. So our lab has run Kaggle challenges to identify models that can basically turn these images into an embedding, and these embeddings can then be used in, for example, the multi-scale maps that we built in the CM4I project, where we integrate this image data with the PPI data, or in other projects. And here's just another study where we contributed with these embeddings to revealing molecular grammars underlying nucleolar suborganization. So when we look at the first initial data set of the CM4AI project, first initial image data set on breast cancer cells, and we embed this with such an AI approach as this DenseNet that we have used here to embed all the Human Protein Atlas images, then it looks like this. We see how in this UMAP, the features spread out different fields of use or different images in the embedding space. And you can see that we can set this embedding space separate different subcellular localizations. For example, here we see mostly the nucleoplasm. Here we see a lot of cytosolic labeling. And we see also some changes when we just compare the different treatments. So today you get the chance to work with this data set and explore how to convert it into an image-based embedding. And you're highly encouraged to try to further play with this data and see what you can find out here, how protein localization, morphology, many more aspects change. And with this, I'm at the end of my presentation. I want to acknowledge the lab, many very collaborative, exciting people, our funding sources, of course, the British AI project funded by the NIH that makes this work possible. And yeah, wish you a lot of fun.